with that, I want to introduce our speaker for tonight, Dr. Justin Chaffin. Where's Dr. Chaffin? Dr. Chaffin, really from a standpoint, it wasn't that many years ago that Dr. Chaffin was each like you guys, and was here as a student from Bowling Green at the time. Am I right, Justin?
Yeah, Bowling Green.
And then, had the experience as a student. Came back that I wanted to do an REU, became an REU here. Worked with Dr. Kane, I think, and was very much involved with the nitrogen and phosphorus work. Went on, graduated from Bowling Green, went to University of Toledo with Dr. Tom Bridgman, who you'll find up on our board over there someplace. Dr. Bridgman, I gave a scholarship to Dr. Bridgman in maybe 1989 up here as a student at that time. Got his PhD working on the nutrient issues. Justin then worked for Dr. Bridgman exactly on the problem that we had the press briefing about today, and your presentation on the monitoring and the different challenges. And now the two REUs that are working for you, you guys did great jobs today. This was very, very good. So I think, Justin in many ways is like, somebody you could look at and say, boy, I could do that. Same thing with Matt Thomas. Matt Thomas was a student up here in about 95, am I right, Matt? And took Dr. Hogarth's course, it's very common to have this experience, not necessarily going to bring you back to Stone Lab in any kind of a way, but what you're likely to find is the people that are sitting around you right now are very likely to be your colleagues, your peers, for the rest of your life. People that I was up here with back in 1971, I still interact with them. They're, now, one guy's probably one of the top 3 knee surgeons in the country. A couple of them became dentists, but then others went on to more meaningful jobs. The biology and that kind of stuff. But just good friends. So it's likely as you look around the room, you're probably going to know these people for the rest of your life. Interesting opportunity, and take advantage of it while you're here. Today, Dr. Chaffin's going to talk to us really about the implications for dual nutrient management, and sort of looking at the nitrogen and phosphorus. And some of you are working on that right now, and it's what is basically your PhD work in many ways.
Right. Yeah.
Okay. Very good. And Justin, as you start, you may want to talk a little bit about growing up here in sort of north central Ohio, and what got you into this? Why you even came to Stone Lab in the first place?
So my hometown is in Collins, Ohio, which nobody knows, but it's near Norwalk. Maybe nobody knows that either. But there's a raceway there, and I'm kind of near Sandusky. So I'm from Sandusky area. I grew up. Parents' house was 5 or 6 miles south of the most southern point on Lake Erie. This pointer won't work. I'll just use this map. Well, anyway, see the most southern point on Lake Erie? I grew up about 7 miles due south of that, and parents had a boat, and I was always on the lake, mostly walleye fishing and then perch fishing. In high school, I knew I wanted to do something with not business. So if you're not going in business, you're going in some sort of science field. And then I started off as a nutrition major, but one of the classes I had to take for my nutrition major was the introductory to biology, which is like at Bowling Green, it was like everything from the cell up to people, and that really got me interested in the environment. dusty biological field station, but they told me they wanted a graduate student to help them collect fish. And remember one day at Firelands College, I did my first 3 years of Bowling Green at the Firelands campus. I took a flyer off the bulletin board for the Field Biological Survey Network or whatever, and that's how I found Stone Lab. And then I applied the next day, and that's how I got started up here. So my presentation.
History.
Yeah. So my presentation today is really going to be a continuation of what we heard from this morning, and a lot of my research, well, the bulk of what I'm presenting was conducted during 2011, which turned out to be the biggest bloom. I didn't plan on it being that way, but if you look at the map over there, the one in the lower left, all that red is cyanobacteria. Now I'm going to describe factors that cause that bloom, and then what happened into it. So Kevin?
Third time?
Alright, so the outline today, I'm going to first introduce you to the idea of dew nutrient management. And then I'm going to discuss the factors that caused the 2011 bloom. And then we're going to look at, inside that bloom, looking at nitrogen, phosphorus, and the cyanobacteria that made up that bloom. And then we're going to discuss some future implications for future blooms and current and future research. But before I do that, I just want to highlight my 2006 REU. This was so Doug was 4 years ago. The experiment, we had mud, and then mud with mayflies, and this one had mayflies, a lot more mud, a lot more phosphorus, and it was published in the Journal of Great Lakes Research in 2010. So the message is, you might not get it published right away, but keep with it, and eventually you could get it published. And it turned out to be a good building with Doug, so good connection.
You published your REU research.
Yeah. So back to algal blooms. These blooms are a global problem. Have blooms on every continent of the world, basically except for Antarctica, too cold there. And the main reason for the blooms are human activities increasing nutrient loadings to lakes. They have blooms in China, as shown here in the picture, blooms in Europe. This is the 2011 bloom off of across from the Lake Erie Center in Oregon, Ohio. And also to note, this is Microcystis, and below it is another cyanobacteria called Lingbia that grows on the bottom. So we have these blooms that are occurring all over the world. It's just not an Ohio problem or just a Lake Erie problem. Problems associated with these algal blooms. Most of these algal blooms are cyanobacteria. We often use the word harmful because they can produce toxins. During these cyanobacteria blooms, there's a low diversity of phytoplankton, so the diatoms and green algae, the good algae die away, and you end up with nothing but cyanobacteria that zooplankton really don't like to eat. You can see all this large biomass, it will die and start to smell, and water will taste bad. As that biomass dies and sinks to the bottom, it can consume oxygen. And you can imagine if you live on this lake, your property value may decrease. There's also big negative economic impact. People don't want to go boating or go fishing when they see these blooms. So work that was done in the mid-60s and 70s really showed that high concentrations of phosphorus are required. This is an experiment done by David Schindler in the late 60s. And this lake is a lake in northern Canada. In the center of it, he divided the lake. Top half you had a carbon, nitrogen, and phosphorus, and it developed a thick cyanobacteria bloom. And on the other half, on the lower side of the graph, carbon and nitrogen added no phosphorus and no bloom developed. So experiments like these and other experiments at that time really hammered down what became known as the Phosphorus Paradigm. And it states that phosphorus concentration regulates the amount of algae biomass in a lake. And that Phosphorus Paradigm also states that any long-term limitations of carbon or nitrogen will be alleviated because there's carbon and nitrogen in the atmosphere. There's no phosphorus in the atmosphere, so if a lake runs out of phosphorus, it's not going to get any more from the atmosphere. If a lake runs out of carbon or nitrogen, the algae can tap into the atmosphere. And within a lake and among lakes, the amount of algae biomass closely correlates to total phosphorus rather than total nitrogen. And it's been shown that if you decrease the amount of phosphorus that enters a lake, you'll get less algae biomass. And this was shown on Lake Erie in the mid-1970s when we improved our sewer treatment plant. The amount of phosphorus going in the lake decreased, and the blooms went away. However, recently there's been a erosion of the phosphorus paradigm, and this paper published in 2008 by Lewis even uses the word erosion of phosphorus paradigm in the title. And then in 2009, a group of authors published in Science, Controlling Eutrophication, the Need for Nitrogen and Phosphorus. And together they kind of came up with this dual nutrient management strategy. And this calls for a regulation of phosphorus and nitrogen, not just one, but both of them. And some evidence for this. I'm going to hit on this near the end of my presentation, but humans have doubled the amount of available nitrogen by anthropogenic nitrogen fixation. So back in the 1900s, compared to now, the amount of nitrogen that's available is more than double what it was before humans really started doing stuff. And also, our blooms we heard about today, this morning, we have Microcystis, which is a non-nitrogen fixing cyanobacteria. It cannot use atmospheric nitrogen. And blooms of Microcystis are spreading around the globe as opposed to nitrogen fixing cyanobacteria. And this could be an indication that there's too much nitrogen. And a lot of lakes around the globe, the amount of algae growth is being stimulated by nitrogen. If you add phosphorus, nothing might happen. You add nitrogen, increase growth rate. And if you know much about the nitrogen cycle, you know that nitrogen can be lost from a lake. And this process is what's called denitrification, where nitrate is reduced to N2, and then the N2 can go into the atmosphere. Well, this process does not balance humans putting in more nitrogen. However, many limnologists disagree with this dual nutrient management strategy, and they say it should solely focus on phosphorus, and that nitrogen abatement is not necessary. And this is highly debated in literature. This is just a short table of recently published papers on the left saying we need management of phosphorus and nitrogen. Columns on the right saying only focus on phosphorus. And as David Schindler and Bob Pecky pointed out in their 2009 science paper, they say more nitrogen data is needed. And as I was starting my PhD, I was looking for possible topics. And when these lists of science papers came out, I thought, hey, this is a possible opportunity for research. And I believe that Lake Erie can provide insights to this dual nutrient management issue. So this is a graph collected by my graduate advisor Tom Bridgman at the University of Toledo, and this shows concentrations of nitrate in Maumee Bay on the left, and in the center of the Western Basin on the right. This graph shows two basic trends. If you look at the scales in Maumee Bay, you have 500 to 0. These are in micromolar. In the center of the basin, 100 to 0. So we get less nitrate as we move away from the river. And then you look at across the season, each dot represents any data point, like between 2009 and between 2002 and 2009. And we see a decrease in nitrate. And, a lot of times in Maumee Bay, we're getting below detectable limits by mid-August. So this allows us to compare what's happening to the cyanobacteria in high nitrogen waters versus what's happening to the cyanobacteria in low nitrogen waters. And we also have cyanobacteria in Lake Erie. And these blooms, for the most part, occur in August when nitrate is really low. And we have a lot of variation of Microcystis in Lake Erie. This is west of West Sister Island, and most of my work was conducted here in 2011, which was by far the worst bloom. So now I'm going to look into some of the factors that help explain why 2011 was a very bad year. Well, we heard today from Richard Stump that you look at the amount of phosphorus that enters the lake from spring through March through June, and we stimulate a lot of cyanobacteria bloom. Well, 2011 had by far the most phosphorus loading. And if you missed the prediction, the bloom for 2013 is projected to be somewhere around 2003. So we have a lot of phosphorus loading coming in during the spring. And this graph shows that most of the phosphorus loading occurs during heavy spring storms. So if you just focus on the solid lines, the blue line is discharge of the Maumee River. When the solid blue line goes up, it means it just rained, more water is getting into the river. And the red line is the amount of dissolved reactive phosphorus entering the lake through the Maumee River. So every time we have a storm event, phosphorus loading goes up. You have another storm event, phosphorus loading goes up. Storm, and another storm. So it says most of the phosphorus loading occurs during a storm. And then, so following those heavy spring storms, we had a relatively calm summer. And that calm weather during the summer allowed all that spring water that came down the Maumee River just to sit in Maumee Bay and incubate. So this graph shows the residence time of western Lake Erie in days. And this little subset graph shows a typical year. So for a typical residence time of Maumee Bay is normally less than 40 days. So this is the percent of years, and these are the time. Most times, water enters from Maumee River and is flushed out of the Western Basin in 40 days. In 2011, the water entered in Maumee Bay and just sat there for up to 80 days. So we have stormwater that has very high concentration of phosphorus. Now it's just sitting in Maumee Bay. So we have calm water, but why does calm water favor Microcystis? This is an ecology paper by Jeff Hushman. And if you look at, this is how much light is available in a given lake. And this is if the lake's calm or if the lake's mixed. And in calm, turbid waters, Microcystis wins. In clear, mixed waters, the diatoms and green algae win. Microcystis wins in calm, turbid waters because it can float to the surface, as you see in the top left there. It can float to the surface and get light. That's why it wins in calm waters, calm, muddy waters. Diatoms, they're heavy and they'll sink. So if you do a plankton sample and an algae, you let that plankton sample sit for a couple minutes, diatoms sink right to the bottom. So diatoms need mixing. They need ways to get access to sunlight. So that 2011 record-breaking bloom can be explained by large spring storms. Those large spring storms had high phosphorus loading, which in turn fertilized growth. And following the spring, we had a very calm summer, which allowed Microcystis to float and thrive, while diatoms sank and died. So that's kind of like a big scale picture. Now I want to look into what's happening in the bloom itself. And I want to start off by looking at which nutrient limited the growth of algae in western Lake Erie during this big bloom. So for some of the methods, I used experiments which you take lake water and you incubate it with additional nutrients. And I did these experiments monthly, June through September, and in 2010 and 2011, but I'm only going to present 2011 data today. And you measure the amount of chlorophyll before and after incubation. And I did this at 2 sites in Maumee Bay and one in the center of the Western Basin, which is kind of near West Sister Island. And for just like a cartoon to make it a little bit simpler to understand, this is what you start off with. You have your control. So you have lake water in some sort of flask or a beaker or a bottle. Control it, add no nutrients, add phosphorus, add nitrogen, add phosphorus and nitrogen together. This is your initial measurement. And then you allow that lake water to incubate, either in situ or in a growth chamber or somewhere. We allow it to incubate. And if you get phosphorus limitations, ones where you added phosphorus will turn green. If you add nitrogen, nothing will happen because this indicates there's already enough nitrogen here. Nothing happens here. But in this one, you still added phosphorus, so you get green. So this is an indicator of phosphorus limitation. An indicator of nitrogen limitation is you add nitrogen and you get algae growing. Adding phosphorus did nothing. It indicates they have more than enough phosphorus. And sometimes it happens when both phosphorus and nitrogen are so low in the bottle that they need both of them together, and we call this a code limitation. And sometimes they have more than enough nutrients and they just grow like crazy. And we call that no nutrient limitation, or some sort of the growth is nutrient replete. So the start of the bloom was nutrient replete. No response to phosphorus, no response to nitrogen. So the growth of that big bloom was totally unconstrained. The peak of the bloom in the Western Basin in August was nitrogen limited. No response to phosphorus, big response to nitrogen. But also note that we get the biggest response when we add phosphorus and nitrogen together. So the peak of the bloom was nitrogen limited. And when I did this in September 2011, also nitrogen limited. 2010, August and September were nitrogen limited. And last year, nitrogen limitation started in July. So this nitrogen limitation seems to be pretty common in western Lake Erie. And if you just look at if you don't like data, sometimes pictures help. You can see what happens when you add nitrogen to these flasks. And also monitored the amount of nitrogen, phosphorus that was ambient in the lake water. And in the early summers of 2010 and 2011, very high nitrate, very high dissolved reactive phosphorus, big bloom. And growth was not constrained by phosphorus or nitrogen. But then by late summer, note that nitrate has been depleted. And ammonia in western Lake Erie is pretty much below 2 micromolar at all times. So when nitrate runs out, the algae run out of nitrogen and they become nitrogen limited. So I showed that nitrogen limits phytoplankton growth during 2011, during this big bloom. Now I wonder, how did the cyanobacteria community respond to this? So for this, I did a big field survey during 2011. Again, wasn't planning on doing the biggest bloom on record. I was planning on doing this field survey in 2011, just happened to be the biggest bloom. So I had 10 sites in Lake Erie that I sampled from June through September, and these sites were from Maumee Bay and Toledo all the way out to Avon. And I collected two additional samples in October when MODIS satellite showed there was a big bloom out there, so I ran out, got additional samples. And each sample site collected water column samples for nitrogen, and broke the nitrogen up into many different forms. For phosphorus, total phosphorus, DRP, dissolved organic phosphorus, identified and quantified cyanobacteria by biovolume. Microcystin is the toxin that we're most concerned about in Lake Erie, the toxin produced by Microcystis and other forms of cyanobacteria. Chlorophyll A, and a whole bunch of other measurements. And here's the 10 sample sites. Well, I guess I have one in each river, then 8 in the lake. But I have one in Maumee River, Maumee Bay, Western Basin West, Western Basin Central, and Western Basin East, Sandusky River, Sandusky Bay, Sandusky Subbasin West, Sandusky Subbasin East, and a Central Basin site. And for my next couple of graphs, the data is going to be arranged from river, bay, basin, river, bay, basin. So first, just start off looking at the nitrogen concentrations. There's a lot of data up here, but I'm going to point out the highlights. On the top graph, I have nitrate, and note the sites, Maumee River, Bay, and Western Basin, Sandusky River, Sandusky Bay, and the Subbasin and Central Basin, and the different colors indicate different months. So the top graph is nitrate. We note that in August, very low nitrate in the Western Basin, and Sandusky Bay runs out of nitrate by July. Yeah, Sandusky Bay out of nitrate by July. Ammonia, you note the scale. This only goes from 0 to 10, whereas nitrate went from 0 to 625. So low ammonia in all samples. And the bottom graph shows dissolved organic nitrogen. And this ranges from between 15 and 60 micromolar on all sites. And we see a general decrease from river, bay, into the basin. And a lot of times dissolved organic nitrogen is overlooked by freshwater monitoring programs. And there's many different forms of organic nitrogen. but quite a few of them can be used by algae. So something we might want to start paying more attention to is dissolved organic nitrogen, especially late in the fall when they run out of nitrate. And also to point out that the 2 samples that I collected in the central basin, or in October, the blue bars, were in the Sandusky subbasin east and in the central basin, very low nitrate. And I'll bring that point up again later. And those 2 sites were only sampled in October. And so this graph shows the amount of cyanobacteria, total cyanobacteria, and note the scale is a log scale. In the western basin, the peak of the bloom occurred in August, while in the Sandusky subbasin, September and October were the peak of the bloom. And in Sandusky Bay, the amount of cyanobacteria exceeded what was in the lake. Now, breaking those bars down by species, we find 3 main species of cyanobacteria in Lake Erie. The first one is Microcystis, the second one is Plankothrix, and the third one is Anabaena. So looking into the July samples, Maumee Bay and the western basin, mostly Microcystis. Same thing with the subbasin sites, mostly Microcystis. But Sandusky Bay, a lot of Plankothrix. So Microcystis, dominant cyanobacterium, most of the lake in July. Plankothrix, which is the filamentous one in Sandusky Bay. And Anabaena, which is a nitrogen-fixing cyanobacterium, was less than 20% in all samples. And also importantly, all 3 of these species can produce microcystin. Looking into August and September, see an increase in Anabaena in August and September during when nitrate concentrations were depleted. We still have a lot of Plankothrix in Sandusky Bay. And this October central basin bloom was more than 95% Microcystis, even though nitrate was almost depleted. And the concentration of the toxin microcystin, see that the highest concentrations matched the peak of the bloom in the western basin. And Sandusky Bay always has a lot of microcystin because of Plankothrix. And then at October bloom in the central basin, very low microcystin. And I think it's because there was almost no nitrate available. So I have all this data. I just want to look for simple correlations among the parameters. And these are Pearson R values. And I have significance indicated by if 1 star is p-value 0.05, 2 stars is 0.01, and if there's no star, it's 0.1. And anything above 0.1 I consider non-significant. And I removed Sandusky Bay from this regression because it's completely different from the rest of the lake. So first thing to point out is high correlation among the particulate nutrients and the cyanobacteria and chlorophyll A. This is not surprising because a lot of these particulate nutrients are in the cyanobacteria. But we do get correlations among ammonia and dissolved organic nitrogen with Anabaena and microcystin. Another reason why maybe we should start paying more attention to dissolved organic nitrogen and ammonia in lakes. And then finally, if you just look at a simple regression between Anabaena biovolume and microcystin concentration, you see that Anabaena explains 77% of the variation in microcystin concentration. So we see a decrease in bioavailable nitrogen throughout the summer, and that low nitrate indicates that nitrogen limitation is probably lake-wide. At least it was during 2011. And again, here's the picture showing what happens when you give cyanobacteria bloom phosphorus in August. Nothing happens. You give it nitrogen, and then they grow like crazy. And dissolved organic nitrogen can contain the largest proportion of the nitrogen pool in late summer. Again, like I said earlier, often overlooked in freshwater monitoring programs, and many of these dissolved organic compounds are bioavailable. And we saw an increase in the nitrogen-fixing cyanobacteria, Anabaena. However, we did see Microcystis was able to persist in these low nitrogen waters, and it's more likely because they were just being transported with water currents and wind. And that October Central Basin bloom was 95% Microcystis, even though there was very low nitrogen. So one of the conclusions I think I can make is that Microcystis, or Anabaena, was a likely toxin-producing cyanobacterium in 2011. However, this is just a correlation, and correlation does not equal causation. But also, as shown, Anabaena is capable of using atmospheric nitrogen, whereas Microcystis can't. Microcystis requires some sort of combined nitrogen for growth, and many lab experiments confirm that Microcystis will stop producing microcystin during nitrogen-limited conditions. So if we have nitrogen limit conditions, then we have Microcystis, it's not going to make microcystin. If we have nitrogen limit conditions, then we have Anabaena, then Anabaena will still be able to make microcystin. So bring back to the topic of the nitrogen abatement. It has a lot of support recently, and it calls for management of both phosphorus and nitrogen. However, I think the data that I collected in 2011 show that low nitrogen concentrations will not affect microcystin. That low nitrogen will likely result in more Anabaena. So if we do reduce the amount of nitrogen getting in the lake, we'll just shift the bloom to Anabaena sooner, and it will still be able to produce microcystin. Well, what about if we increase nitrogen, right? That'll be the other thing to go about. Well, that's exactly what we've been doing. So this graph shows human input of nitrogen into the ecosystem. This is on a global scale. This is through 1990 through 2000 with the data and projected. So we've been adding more nitrogen to the system. Well, so some of the research that I'm working on now and hope to in the future is to understand what will happen if we continue that upward trend of putting more nitrogen into the system. And I have a hypothesis that increasing nitrogen will likely increase cyanobacteria biomass and toxicity. And on the toxin side of the argument, this is a microcystin molecule, the toxin, and the ring structure is based on amino acids. And if you remember from general biology, amino acids are nitrogen-based, and microcystin LR is 14% nitrogen by mass. And most of the other cyanobacteria toxins out there are also nitrogen-based. So saxitoxin and antitoxin are also require nitrogen. So Microcystis cannot produce microcystin without some sort of combined nitrogen. But if we have Anabaena blooms, the Anabaena will also use a form of combined nitrogen if available. So if the system runs out of nitrogen, they will switch to end fixation, but if you put more nitrogen back into the system, they'll take advantage of whatever form of nitrogen you give them. And since nitrogen fixation is so costly energetically, if you give them more nitrogen when there have been nitrogen fixation, they'll increase their growth rate. So I think that if we're loading more nitrogen into the system during the bloom, we just may be throwing more fuel into the fire. So I just would like to thank a couple of my lab mates that are really helpful. Sarah Panic, who's a master's student that graduated last May, and Courtney Mobilens, who's an undergrad who just graduated this May, and our lab tech Peter Bakir, who just moved out to California. This is in Sandusky Bay. You can see Cedar Point there. So I was the one that drove the boat around, and they untangled the YSI cords for me. And I have a whole list of other acknowledgements. My advisor, Dr. Tom Bridgman, Darren Beatty was on my committee, Mike McKay was on my committee, who spoke 2 weeks ago. A lot of other people at the Lake Erie Center and University of Toledo's main campus. I also used Stone Lab's boats there in 2011. And funding sources, a lot of this was funded by the Ohio Lake Erie Commission. So if you see those little license plates that have the little lighthouse that say Lake Erie, our Great Lakes, money they raise from you buying license plates goes to fund projects like mine. Ohio Sea Grant providing ship time. National Science Foundation provided me with a teaching fellowship for 2 years, and the International Association for Great Lakes Research for a student scholarship. So, again, it froze on the final slide, so good timing.
Thanks, Rick. Questions for Dr. Chaffin?
Yeah, it's great study. So what are the sources of all of this phosphorus and nitrogen in the farms?
So most of the nutrients enter, first to say for Lake Erie, most of it is agriculture, coming from the Maumee River.
Yeah.
For nitrogen, it can be other sources of manure, or basically it's all fertilizer practices. Fertilizer, whether it's commercial or manure, that's where the excess nutrients are coming from. And when it rains, it's carrying it off the field and into the river.
So what happens with all your great work? Where does that go? Does it get any action plan or changes in regulations? What happens?
Well, we hope we can make some sort of recommendations, and right now we're focusing on lowering phosphorus load. So if we bring the total amount of phosphorus load that come from the rivers by about 40%, we think we should be able to reduce or remove the blooms in Lake Erie.
So how do you do this? Do you call it Scott's fertilizer and say put less phosphorus in it, or what do you, I mean,
well,
I know I'm oversimplifying it, but
Scotch just
how does
removes
something
their
like that happen?
phosphorus fertilizer.
How do you reduce phosphorus?
It has to be, a lot of it has to be done through changes in agricultural practices.
Okay.
So it's not necessarily how much they're putting on their fields, it's when and how. So if they apply fertilizer right before rainstorm, or if they broadcast, it all gets washed away. So it's mostly how and when farmers will fertilize their fields.
There are some good points. You brought up Scotch. As of January 1, because of the work done up here and at the Phosphorus Task Force in Ohio, Scotch has removed phosphorus from their lawn care products. With the exception of their starter fertilizer. So if you're just planting a lawn, you're still going to have some phosphorus in that. But if you went and you looked at the 3 numbers on the back of a bag at Lowe's or there are a number of other practices like that. The Farm Bureau is backing this, and that's incredibly important because with the scientific community, they're very likely to take action based on things that we say up here. But the agriculture community is actually more, it's sort of the trusted spokesperson. The Farm Bureau tells them to do something, makes a recommendation, probably more likely that they will do that than a recommendation coming from us up here at Stone Lab or from Ohio EPA, for that matter. Just that trusted. So that's a good point. Quick question for you. The technique that you were using, I'm just wondering about the concentration of both nutrients and microcystis and even the toxin. When you do your toxin concentration, is that also based on a whole water column sample, or is that just the surface sample?
So, toxin, the nutrient data, and the biovolume data, all the data presented was an integrated water column sample. And I remember I had the comment, some said, well, that's not a very high microcystin, it was only 8. But remember, those samples were integrated over the entire water column. And if you assume that, most of the microcystin is concentrated right at the surface, and you do back of the envelope calculation, assuming that all the microcystins that in that top centimeter or so, concentration has been probably over 1,000 parts per billion, maybe even higher.
I often think that for the nutrients, the water column probably makes sense. But for the toxins, I often wonder if we ought to be thinking more about right at the surface, or sometime just doing a composite maybe of the first meter or something. Obviously, if you have mixing, it's going to be less important. But a lot of times it is right at the surface. It's something we ought to think about anyway. But also, if you're doing all your comparison of concentrations, Western Basin, where you might have a tube sampler down 8 meters, and Sandusky Bay, where you might not be able to get more than 3 meters, or something like that, that could have that same dilution effect. And so it's hard making those kinds of comparisons. But I really think you're right on. There's just more and more data popping up all the time. That I think we're doing the right thing by phosphorus limitations. That's clearly the right thing to do. But I think both, we could figure out ways to limit both, we'd be even more effective. Any other questions, or Doug?
So, you had a good suite of field and lab experiments, and both are really good for different things, and I've often done that end. I think the hardest, but some of the most useful, is in the middle with mesocosms, where you can control some of the factors you want to, but also have some of the more natural parts of the environment. Now that you're up here all the time, you want to run some mesocosm experiments, you can ask CRISPRs.
It would be nice to get I like what they have at Kellogg pilot station, where they call them liminal crowds. But then with those in Lake Erie, you're only stuck with experimenting with water that's right next to it. And if you want to study cyanobacteria every year, and their mesocosm, you have to hope for a bloom to get out here, because you can't just transport 100,000 gallons of water from Maumee Bay over here. That's why you got to go small scale in a big lake.
Thanks very much, Dr. Chaffin. Why don't we take about a 5-minute break, allow people to talk to us about a number of issues or programs, things that are happening at Ohio EPA, and I think from a science background and from the standpoint of the students in the group, Director Nally is a perfect person to be listening to and to be asking them questions afterwards. And as we start this, Scott, I'd like you to help the students understand how you made decisions challenges, choices along the way, majors, those kinds of things. Director Nally grew up in upstate New York in the Finger Lakes area. Pretty diverse background at that point, very interested in the sciences, but went to NC State for his, North Carolina State for his bachelor's degree in kind of general sciences. He's been at, I should probably start by saying, he's been director of Ohio EPA since 2011, January 2011.
2.5 years.
Yeah, I believe you started like in January 10th or 1st or something like that.
Sworn in at midnight on the 10th, 1 minute after, and was in the office 5 o'clock the next morning.
You have that reputation, I understand, for being a real early person into the office. Prior to that, he'd been at the Indiana Department of Environmental Management, which is the Indiana version of Ohio EPA. And he'd been there as the #2 person for 25 years. So he's been basically in the compliance regulatory end of management for 27.5 years, something along that line. So a good career. But I got to tell you a little bit about his background beyond. After he got his bachelor's degree at NC State, I'm real curious to hear how you picked then to get a master's degree in biology at Wyoming. And then from Wyoming, he went on to work on his PhD at Purdue. And Director Nally's expertise was really in the tracking and preventing and understanding the biochemistry of foodborne pathogens. And so he was working on his PhD at Purdue on foodborne pathogens within eggs. At the time, some of you may remember, this is quite a few years ago now, but you might have heard Rose Acres had a salmonella outbreak where it was harming people from the eggs, and they were able to, with Director Nally's help, trace the source of the pathogen all the way back to the individual farm that the eggs had come from. And at that point, his expertise, the things that he was working on for his PhD, made him so valuable to Rose Acres that they literally pulled him out of his PhD. He probably offered you a little more than you were making as a graduate student.
Which didn't take much back then.
And anyway, so he left his PhD to take that job and work on that issue with Rose Acres, and then shifted and went over to Purdue Farms, which is basically a giant poultry industry, doing exactly the same thing, foodborne pathogens or preventing foodborne pathogens, but not in eggs, but in the chicken that we would eat, that we would buy in a store, and sort of how do you prevent that based on the processing, and when you do have problems, how can you trace it back and figure out where the problem occurred, solve it, and end that. Today, we're here at the 40th anniversary of Ohio EPA.
Right.
And so you're going to tell us a little bit about Ohio EPA, the things that are going on. And I've asked him also, if in addition to talking about, sort of his background, how he made these choices between upstate New York and NC State, Wyoming, and Purdue, to talk a little bit about opportunities for students, internships, and things like that at Ohio EPA. So please join me in welcoming Director Scott Nally, Director of Ohio EPA.
Well, it's been a long day, but I'm glad to be here today. And I think a rule of thumb or something to take home is oftentimes the path that we travel is serpentine. I mean, if you were to ask me as somebody growing up in upstate New York on a dairy farm and out running around and getting in all sorts of trouble, would I end up at Ohio EPA as the director? I would have said no. Matter of fact, if you'd asked me maybe 10 years ago if I'd be here, I'd still say no. And so oftentimes we take serpentine routes, and it is decisions that we make. You get the forks in the road, and as Yogi Bear says, take it. So the idea is you get to the fork and you take it, you make the best choice that you can. NC State, I ended up there for two reasons. One, I was recruited out of high school and had multiple choices to go to, and that was one of the better schools of the choices. So I went there predominantly on a sports scholarship, and had the ability, because academia and NC State is well known for its agriculture and well known for its science program, and it was an honor to be there. I ended up staying in a research station, and as you talk about things that get you excited, Justin, you end up living on a research station as an undergrad. Your eyes get opened up as you're working on plant path and plant fizz, and then you're out in the fields doing some tobacco research on nematodes. And then you start meeting folks, and next thing you know, you're making a career choice to go to the University of Wyoming, of all places. And then there you're doing postmortem interval studies, working with FBI. We had the first body farm prior to Tennessee having a body farm. So we were looking at using predominantly microbes, but also insect, flies and beetles to determine time of death and oftentimes location of death. And one of the projects that I worked on brought me to Indiana. There was a murder case where some bodies were dropped in the back of a trunk. The trunk had sat pretty much the month of July and August in Indiana at a 7-Eleven, and they were trying to speciesate the fly maggots in the trunk. Called me in from Wyoming to Indiana. I was able to help the detectives kind of connect the dots, got some people thrown in jail over it, and that got me to Indiana, of which I was working on my PhD at Purdue University, again, choosing the school based on the program that I wanted, irrelevant of what state it was in. Oftentimes we're just fleas, we just need a ride. Back then, I just needed a ride. So ended up in Indiana there, and then quickly got sucked about a year and a half into my program into Rose Acres and then Purdue Farms, the other spelling. And again, trying to use some of the different skill sets that I had learned. One, looking at cause and effect. Two, being able to identify, go through taxonomic keys, diconic keys, work through all of that, and then to be able to connect the dots. So you end up working on Salmonella and Campylobacter and Listeria, and we were developing, again, out of the box, live vaccines. We were doing Phosphorus Salmonella. Right at this point, I never get food sickness because I've self-injected by mistake multiple times in my hand. It's amazing how swelled up your hand can get with an autogenous vaccine when you oil immersion and you panic and step on the button while you stab yourself in the hand and keep pumping it in. Of course, you're a kid, you know, you're 20 years old, 21. But you just get all that background, and then you end up, when you get a calling, in this case from Indiana, from Governor Daniels at the time, who I'd met when he was campaigning, and said, why don't you come over to IDEM and help us kind of work through and clean up state government? We've got some issues. And I said, really, are you kidding? I don't want to take an 86% pay cut. So I went up there to tell him no, and about an hour later I was doing it. And then I thought eventually I would go back into the private sector. Well, Mitch and John, the current governor of Ohio, John Kasich, were good friends. They knew each other in D.C. So I got traded for a first-round draft pick. So, and I'm sure to be named later, since now Mitch is the president of Purdue University. So it's a small circle, and as Doc said earlier tonight, the people that you're rubbing elbows with, you'll know forever. He's right. It's amazing how small the world is and how oftentimes you bump into people and strange places and strange times. So sure enough, here I am, and been over at Ohio EPA now since January of 11, so 2.5 years. And we met with some of the students here earlier, and you do get better at presentations. I've been on the job about 910 days, give or take a day or two, and I've done 948 of these in 910 days. So you do get a little better at it, at least I think I do. It's a challenge oftentimes when the topic matter changes multiple times in a day. So you're talking water in the morning and solid waste at lunch, and then a CSO issue in the afternoon, but you get to juggle it and get pretty good at it. So what I thought I'd do is kind of walk you through what Ohio EPA is. And then we always hear the doom and gloom, so I thought I'd kind of put some things in perspective. We are one of 3 agencies that were started originally when Nixon created US EPA, which is why we have EPA in our name. So Ohio EPA Clean Water Act just celebrated its 40th year. We just celebrated our 40th birthday. So in our logo, that reflects that. And compare that to another agency, for example, IDEM, Indiana Department of Environmental Management, doesn't have EPA in its name, and it just finished celebrating its 25th year. So we've been in place for 40, IDEM's been in place for 25, and you've got the other 48 states that are somewhere in that range. Illinois and California were the other 2, and they have EPA in their name. So that was the first 3 agencies in the country that were at the state level. And then those 3 agencies have the largest delegated authorities. So oftentimes through the Clean Water Act or the Clean Air Act, I am then the signatory for the state on issuing water permits, wastewater permits, and air permits, for example. And I'll go through some of that. But a lot of my counterparts in other states don't have that delegated authority that comes out of DC. So I wear all kinds of badges. They got GPS. Since I sit at a cabinet level position, I kind of got to know where I'm at all the times. If I'm MIA for about 6 hours, the cavalry does come running on occasion, so I got to make sure I check in. So it does come with a lot of responsibility, but it's been definitely a great ride. I'm hoping to ride it out for the next year and a half, and hopefully Governor Kasich will, he's already made the announcement that he'll run again. Hopefully he'll win. And I'm assuming I've been doing a pretty good job, so maybe I get to do this for 4 more years, but I'm getting a little tired. The typical day I left the house this morning at about 4:45. That's pretty standard. But we just finished our budget. Governor Kasich signed it on Sunday, and that's been the culmination of a lot of hours. Just in the Ohio EPA component piece of it, we had our standard budget that we presented, which is several $100 million annually, and then the amendment pieces where I was trying to make changes. We had teed up 27 amendments, of which I was successful to get 26 of them. So 26 out of 27, which makes me roughly 49 out of 50 over 2.5 years, and that's a heavy lift. So my legislative liaison teams and a lot of my chiefs, water chief, air chief, drinking water chief, we spent a lot of face time with the legislators, giving them an education so I could get the support to get things through a traditional Congress that might not necessarily be sensitive to the issues that we wrestle with, for example, up here at the lake. So a little bit more about Ohio EPA. Again, that's our logo. These are some of the things we do. I've got several thousand employees, plus contract employees, probably in excess of 3,000 total between contract employees and state employees. And they range anywhere from accounting to geologists, to chemists, to modeling folks, biochemists, toxicologists, and that's actually just my senior staff. And then you work out into the program area. I have programs that do Class 2 and Class 1 injection wells, where we look at geology and seismic reports. That happens out of our agency. I've got staff that write permits, both water, wastewater, and air, solid waste, RCRA, hazardous waste. Remember, Ohio is the 4th largest manufacturing state in the country, and with that comes a lot of legacy issues. So we have a lot of facilities that, as Ohio made a downturn several years ago, become abandoned, and now they've got some pretty juicy chemicals either on them or under them, and we've been going through cleanup on it. But you can take that all the way back to pre-regulation. We were at the hub of a lot of our major steel industry back 100 years ago. We were all over the Manhattan Project. There's a beryllium site not that far from here. There are several federal facilities located across the state where we did enrichment of uranium. We're in the process of cleaning those up. So we're turning somewhere in the neighborhood of about 85 of these brownfield sites to a standard that is either residential or industrial, taking it from its contaminated site down to one of those, and we're doing about 80 of them a year right now. There is a significant backlog, but obviously that's one of the budget issues that we juggle. There's a list of, I think I have 2 slides on this, a couple of things that we do, obviously, a lot of monitoring. I was talking to some folks before. I know a lot of you are doing some lab work. We have a fairly decent sized lab down just in Columbus, just outside of Columbus. We do water analysis there. We do ultra low phosphorus analysis there where you can't even use tap water because even the residual on the glassware comes up as noise in the system. We do a lot of fish tissue work there. We do a lot of soil, a lot of metals work. Pick equipment there. We have it, AAs, GC mass spec, any of the other analyzers, HPLC, a lot of different microscope work as we look at toxin work. We do a lot of the toxin analysis, which is why we then partnered up with Stone Lab to help us. There was a significant amount of effort and time to get the samples down to Columbus within the holding time, so it just made strategic sense to partner up here and do the work up here since the samples were taken up here. But remember, we sample all across the state, for those of you that heard us talk earlier today. So we're sampling Ohio River all the way down in Cincinnati. We're sampling, pick a lake across the state other than Lake Erie. So Grand Lake, St. Mary's, or Buckeye, or any of those lakes that are also having microcystin and toxin issues. So we have actually 23 lakes in the state of Ohio that are of issue like Lake Erie. Major programs, these are some of the various divisions of which I have an air chief. So pollution control program, Bob Hoban-Bosie and his team, they do stack testing to ensure that facilities meet their emission standards. They do all the permitting for all the facilities, and they're on 5-year cycles that we continually ratchet down. And I've got some slides to show some improvements on that, but in a nutshell, Ohio's air and water is tremendously better, and I have data to show that. Oftentimes we get caught with looking through a knothole and everything's always doom and gloom, and where we have been, remember 40 years ago, to put things in perspective, the reason why President Nixon created EPA was because we had rivers, even here in Ohio, that were on fire multiple times, all the way back to 67, 68. So you've got oil sheen, you've got discharges of sewer and wastewater plants that had no controls on them whatsoever, so the water looked like the color of your shirt, and if you threw a cigarette butt into it, it lit. And that was across the state, and that was not uncommon, just like we have currently in some of the third world countries, that if you get a chance to travel around. Various programs, I have district offices across the state, blew through some of these. These are some of the issues that we work with through our air team. Of the air team, direct engineers, permit writers, compliance inspectors, and legal staff, probably about 318 folks, and mix and match on that. Predominantly modelers and engineers. That would be kind of the bell curve if you were looking at the makeup of the MI air team. There's about 8 attorneys on staff. Unfortunately, we're sued both from an environmental group, we're sued from businesses. We're damned if we do, damned if we don't. So if I issue a permit, the environmental community doesn't like it, they sue me. If I don't issue the permit, the business community doesn't like me, they sue me. I get sued about 70 times, 80 times a month. I get pretty thick skin. So I have a lot of attorneys on staff. Try not to say things either that get me in trouble, although at 8 o'clock or 8:30 at night, that's a little difficult sometimes. Just to show some improvements in Ohio since we started recording. And this is actually Ohio being, again, one of the 3 states that is of the oldest. When you look at the number of monitors, which I have in excess of 300 of them scattered across the state, this is actually real data. This isn't modeling data. This is monitoring data. And if you go on our website, the ozone PM2.5 is on 15-minute increments, and it's 15-minute lag, but it's real-time data on our website. So you can scroll across the county, scroll across one of my 300 monitors, and within a 15-minute lag, see what the air quality was off that monitor 15 minutes ago for ozone and PM2.5. But you look at carbon monoxide, the big issue now after the president made his comment last Tuesday and is implementing his carbon capture plan. We've reduced carbon monoxide in the state of Ohio over the last 40 years by 80%. Nitrogen dioxide, 39%. SOx, 71%. Ozone, 19%. Lead, 95%. And then the particulate matter, which originally, when we were measuring this back in the 70s, we were measuring the big stuff, the soot. That was where everybody got the terminology for it. Now we're measuring the real small stuff because we've taken the soot out of the equation. So now we're measuring things that are 2.5 micron and smaller. Significant reduction. So that's our air program, very successful. Our permitting process, incredibly complicated. It's probably one of the more cumbersome and complicated programs that we have. The Clean Air Act is incredibly verbose. It's several thousand pages of federal law. And then when you look at our state rule system, because as we, being a delegated authority for that, we implement federal requirements through state rule or through state law. I probably have 1,300 rules that are just related to air. And they're on 5-year review cycles in the state of Ohio through JCAR. Surface water, George Elmaragi and his team, that's who Jeff and his team's been working with as we've coordinated this joint effort up here at Lake Erie. George does all our NIPIDs permits, stormwater control programs, works with all the cities and towns on their combined sewer overflows. He's got a legal staff that's pretty aggressive and pretty large because we have to deal with a lot of cities and towns as we work through some of the long-term, the 25-year control plans to handle sewer discharges. Yes, it's easy in the basin here to look at the 4 million plus acres and say it's predominantly agriculture, but every time there's a rain event, when you quantify that rain event annually, there's about 8 billion gallons of raw sewage between just a handful of cities on Ohio's side of the basin that put raw sewage into Lake Erie. So we can talk about fertilize on fields, we can talk about animal manures on some of the livestock facilities, which there's actually not that much in the basin there. Most of the livestock ends up in the Wabash, which goes the other direction. So it's predominantly row crop in that 4 million acres. So we can talk about the organic phosphorus and all that, but we still have cities and towns that just because they've entered into an agreement, they're on a 20 and 25-year glide path to address the issues. So when it rained hard yesterday, if you go to my website, there was about 170 million gallons between Toledo and Cleveland that entered Lake Erie, raw sewage. Untreated raw sewage after yesterday's rain event, just to put some things in perspective. So when you're out there sampling today, hope you weren't swimming. Improvements of our water. This is river main stem, which we do TMDL, total daily maximum load studies across the state. But just to put some things in perspective, back in the early 80s, about 20% of our main stems met their intended use. We're now over 89% of that today, meet their intended use. Drastic improvement. So it's nice that we can talk about some of the detail that we are now. Remember, back in the day, we were still worried about raw oils on the water, toilet paper floating down the Ohio River, oil and gas spills and things on fire on the Ohio River that we don't worry about now, unless there's a mishap or a spill, and those are the exception, not the rule, where it used to be the rule. Drastic improvement. Division of Drinking Water, Mike Baker and his team. Every single person in the state should have access to drinking water that meets the drinking water standard. That's our goal. You get that in multiple ways. You get it either from your own private well, which we work with Department of Health and monitor that and then make changes when your septic system is between you and the well, and your septic system is having a negative impact on your well, and then people, we get a little sideways with them. It's kind of funny, but a little sad. But then also you get that through your communities. Several of us are hooked up to a community system, so we ensure that that community system is meeting drinking water standards. If they're in an area where they might come in contact with some of the blooms and some of the issues, we make sure that they have proper controls on, whether it's RO, activated charcoal, other mechanisms, and of course we ask them to sample for it, which costs money and time. But we have very robust program in the state of Ohio. We even administer all the licenses for that. People that operate those systems, other than the single homeowner, has to have a certificate. has a license that says they meet certain qualifications. The biggest difficulty we're having with now are these small systems. They're not big enough to be a city or a town or a township, but they're bigger than just a single home. So it's like a handful of trailers. And then the trailer bought a can plant from a salesman. They put up a can plant. They run the creek through the can plant. Nobody knows how to run the can plant, let alone maintain the can plant. And then about 6 years later, we wonder why we have water issues going into the homes. That's one. The other folks that we have issue with are those that are run seasonally, like campgrounds, where you just don't necessarily have the expertise because they're not operating 24/7 every day of the year. They're only operating a handful of months out of the year, and it's hard for us to get around and inspect. And then the third group we have trouble with, and that's churches. A lot of churches have their own well system. People only go to church typically one day a week, Sunday. So you get 200 or 300 people in a church. If you look at the bell curve of the age, they tend to be on some of our susceptible tails of that bell curve. And they think they don't need to do anything other than bless the water to treat it. So it doesn't take long to get an E. coli outbreak or something along that cost from a church. And that's politically suicide, because then I go find the church. So I write the bishop or somebody a note and says, you owe the state of Ohio $35,000, and oh, by the way, fix the damn thing. And then I get a nasty letter that says, are you kidding me? And then it really gets ugly. But that's the group that we're having some of the biggest difficulty with now. And it is a challenge to fix. Put some things in perspective. My DIMWIM group, when I got on board, solid waste and hazardous waste was kind of scattered all over the agency, so I wanted to make some changes. So I put solid waste with solid waste and cleanup with cleanup. So I have a materials and solid waste group that handles anything that's waste, and then I have a group that handles anything that needs to be remediated. So I kind of re-separated the pie. This group here handles the waste material. That's Pam and her team, Pam Allen, multiple landfills across the state. Got to remember, it was only about 23 years ago that we even put rules in place in the country on how we are to construct and manage a landfill. Traditionally, you would just dig a hole or fill a valley. So you would take a valley, put the trucks up on the hill, dump into the valley. When the valley was filled, put some grass over it and go to a different valley. In the meantime, everything in that valley, what typically runs down a valley might be an old creek. So at the base of that trash someplace is a creek bed, and then every time it rains, it leaches through that trash heap into that old creek bed, and it ends up as somebody's drinking water. We had a bunch of that in Ohio, like every other state. We've cleaned most of those up. Now, over the last 23 years, we actually have engineered landfills. We have landfills with liners and then clay caps and all the various requirements on it. So when we throw things away, we can at least ensure that it isn't going to impact our drinking water. Still have to wrestle with the leachate issue because we haven't been able to come up with caps yet that are completely hydrophobic, but we're getting close. And you still need to be able to breathe on some of these landfills. The landfills actually are a living organism. And the difficulty that we're having in a couple of them across the state, one just north of Cincinnati and one not that far from here, is there's reactions that are occurring in those landfills where they're generating a significant amount of heat. Well, with heat comes gas. So what you have is a landfill that has been closed and capped that is now swelling. And if you can think of it kind of like a hot air balloon, at some point it will pop, and I just don't want to stand next to it when it does. So we're trying to work through the reaction of what's happening now. So I have actually quite a few chemists and biologists working on what that is, and it's not a aluminum dross type reaction. It's something, and we're not the only ones that are seeing it. We're seeing it in several other states, and we're trying to work through what that issue is, but that's what Pam and her team do. And then we look at beneficial reuse out of that group. That group's working on the dredge material, for example, that comes out of Lake Erie. And then we work with the Corps of Engineers on what's the appropriate mechanism or means for disposal. Talking about dumps, 1,300 of them, assuming we could find them and count them, clean most of them up, and now we have 40 targeted engineered facilities across the state. This is an interesting slide. I threw this one in just to make a point. You look at recycling, and you look at when recycling got trendy, and it got trendy pretty quick, but the problem is now, and we spent a tremendous amount of resource on it, we flatlined. And when I came on board in 11, one of the things, actually two points I want to make out of this. One, our solid waste rules and requirements are 23 years old. So we've not kept up with technology. So we've not been driving recycling through state law the way we should. So that's one. The second thing is we've not been able to connect the daggum dots. And that's what's frustrating as hell. Because you have a bunch of folks here, for example, from OSU. By the way, OSU on weekends, you drink a hell of a lot of beer. Just saying. So you got beer drinkers with beer bottles. And I have a glass processor in Zanesville, roughly about 35 miles away. And I have Budweiser in Columbus. You would think that we would be able to connect the daggum dots. So we have people that are generating the glass, God love you, and then we have a processor not that far away, and then I've got an end user all within a county, and we've not been able to connect the dots. So all the glass that's thrown away at OSU ends up in a landfill. The glass processor in Zanesville buys all their glass from Indiana. Budweiser gets all their glass from China. So somehow we have cluster farted that thing around. So now we're going to fix it. We're going to fix it through 592 legislation. We're teeing that up now as soon as our legislators get back from break. A couple of us take a break. We've been going a little quick here lately. And I will tee that up. And what took a decade to pass last time, 23 years ago, we're going to pass it in about 3 months. So hopefully next year, if I'm fortunate to be invited back, I will let you know that we have new solid waste laws in the state of Ohio, and we've finally been able to keep up with the sister states around us. So that's what this, I wanted to put this slide in just to make that point. It's amazing. So that's why we've plateaued. And another point on this, and get to ramble here for a few minutes, I do a lot of traveling. So I spend a lot of time in Lake Europe, and I like to scuba dive. So I've scuba dived in Russia at the Baltic, and I've done the North Sea, and I've done all that stuff. But in doing that, for me, remember, I put dead bodies in fields and looked at bugs and stuff on them. So odd things that we do for fun. And when I'm traveling around, say, Germany, I stop into a landfill in Mecklenburg, Germany, and they're harvesting their landfill. So not only are they not adding, they're adding some volume, but the net of what they're taking out of it is, so when you look at what was a big mound is now a little mound. They still have some trucks coming in, but for every truck coming in, they have 10 trucks going out. They have figured out how to harvest the daggum thing. They can do it, we can do it. So I've partnered up with Swaco, for those of you that are in the Columbus area, and you drive down 71 just there south of town, that big mound on the west side of the road, that's a landfill. And they're on a 17-year glide path to remove that hill. So Project Gemini, you've been reading about it in the paper, we're going to do some really creative things there with Ohio State University's assistance. So we're looking at bringing in some really cool technologies that were incubated at Ohio State, both in Columbus and Worcester. That technology they've been able to go out and get, and now that technology is coming to Columbus, and we will harvest that landfill. So hopefully in the next 4 to 6 years that I'm in my current capacity, I see major strides in that. Then we'll take that on the road to the other 40 landfills. And actually catch up with Europe. Shame on us for falling behind. I talked briefly about some of the contaminated sites we have across the state. I have a very large team that works on that. This is some of the brightest, I mean, all my teams have got bright people in them, but it's kind of like your children. You love them all, but there's some that you love a little more. I'm a nerd. I love this group a little more. And I don't tell them I said that, because then I'll go to their heads. But these are my folks that are the toxicologists. They're the ones that are sitting there and looking at this reams of data. And they're looking at things on 10 to the minus 6, and they're trying to do risk assessment analysis. And they're doing cumulative effects. So it's not just the mercury, but it's the methylated part of the mercury. And it's not just the methylated part of the mercury, but when you're predisposed with something else like PCE or one of the hydrocarbons. So you've predisposed with a hydrocarbon, so now where it was 54 micrograms on the methylated mercury, now it's 12 that impacts you, especially if you're pregnant or lactating female. And these are the folks that sit in these cubes in the dark and work through all this and then come up and give me a recommendation. This is this group here on brownfield redevelopment. And just to give you an example, once again, on board 2.5 years. Cleveland area, when I got on board, remember Ohio in 8, 9, and 10 went into the toilet virtually on unemployment. Unemployment went from 6, 7% to 25%. We had 250,000 Ohioans leave the state. And they walked away from properties. And in Cleveland alone, there are 27,000 foreclosed houses in Cleveland. 27,000. There was. There isn't anymore. We're down to about 12,000 actually. But we've been doing cluster stuff. These are the folks that are engaged with the land bank up there and the mayor and doing some really creative things with asbestos demolition through asbestos NESHAP and going through brownfield redevelopment on the sites so that the sites end up getting cleaned with some really cool technology that again came out of partnerships with OSU. So you've got a group of OSU professors up in Worcester that came up with a glass bead that they can impregnate certain materials in. That glass works as a selective permeable membrane so that when you throw that glass into a bucket of something and water, it absorbs only the something and leaves the water. And then you filter back out the glass beads, run the glass beads into a vacuum, pull out the stuff that you just pulled out of the water, and reuse it. And then take the glass beads that are just glass beads, throw them right back into that contaminated soils again or contaminated aquifer, let it again, just like a magnet, let it go through an osmotic potential gradient, right through the selective permeable membrane, right through the glass, let the glass swell up, pull it back out the glass, and go right back through the cycle again and remediate a site. OSU. Good job, Jeff. But that's the nerdy things that my folks track, and that's the stuff that we're doing. So when we're kicking out 50 to 80 of these a year, and a lot of these are, when you contaminate an aquifer and something that moves literally at a snail's pace, it takes a tremendous amount of time and effort to re-clean up that aquifer. Remember that typically that aquifer is 40, 50 foot underground, so you can't even see it like Lake Erie, and it's contaminated with something that you can't, you're trying to model where you think the footprint of the contamination is. You remove the source, then you got to bioremediate, or you need to pump and treat, or one of the other technologies. That's what my nerds do, and they are incredible, and they do an incredible job. Hazardous waste, just some statistics. Finance. Talked about it earlier this morning when we were talking about wastewater treatment facilities and all that. And these are just a couple of numbers I put out there just to put it in scale. We actually do real projects and spend real money. You're talking 507. I have the ability to bond $1 billion a year. Unfortunately, with the financial status of some of our communities, I leave a lot of that money on the table. So right now, between the two programs, I'm using about $650 million of $1 billion. I have the ability to go to $1 billion. So as much as I want to keep my foot on the accelerator, I'm governed back because of the cities and towns' ability to pay. Remember, you have Mr. and Mrs. Jones that might be retired and they're on a fixed income. So right now they're juggling Medicaid, Medicare, and their house payment. And in the wintertime, they're trying to pay their electric bill to keep the house warm. And if I take a sewer bill from $25 to $300 on some of these projects, they can't afford it. Or they pay that and don't pay their electric bill. So then they freeze to death in the wintertime. So I have to juggle that as we work through. As much as I want to accelerate the cleaning of Lake Erie, that governs me back in Toledo, in Cleveland, in Sandusky, in Fort Clinton. Pick a community there where we have these projects. So that's where the real life piece of this comes into play. I think I'm about done with this. That's my labs. Just to give you an idea of samples, Chris, that's our samples that we do, but are of course much bigger. You get that big, I've given you too much money. Some things that we talked about again this morning. This is the report that went to the governor. We synthesized about 117 pages worth of recommendations. And that was one of two big work groups of three total groups that the teams worked on that you guys heard already this morning. We redid all our signage on the lakes, redid the whole protocol, engaged CDC on it, again, being able to use some of my historical background and contacts. Right now, there's not really a good exposure number. We all kind of guess. We do a little better in guess, but we have certain states that have 10,000 or 20,000 cells per 100 mLs or as an indicator to when they're going to call an exposure or not. Some of us have decided that the cell counts aren't really the right indicator, so we've gone right to the toxin levels. Then the question is, what is the right number? And we've been arguing whether it's 20 or 26 or 32. CDC really needs to help us with that. You're across the board on states, so we've not been very uniform on that. So we've been putting tremendous pressure on CDC to assist us with it. Ohio has taken a position that we're going to be pretty conservative on it. So our first action's at 20, and then actually at 6, and then our no contact is at 20. Some of the states are at 26, or some of them don't even measure the toxin level. They just use 10,000 as the up or down, for example, like Indiana right now, but we've been working with them to adopt this. So this is something that we did about the first 3 months on the job with my teammates, along with some budgets and some other things as you were trying to figure out how to turn on the lights as an agency director. So it's been busy, and it will continue to be busy. We have a lot of issues. Good, it's job security, but we have a lot of issues in the state. You know, as we look through a lot of the brownfields, as we look through helping our communities address some of their issues, as we work with businesses, as permit requirements are continually ratcheted down. There was a standing joke, and I'll get into a little bit of questions here, but there was a standing joke that for the Cleveland area in the 70s to be able to meet appropriate air standards, you'd have to move everybody out of Cleveland. And I now meet that standard that they were trying to meet 30 years ago. Now the standards have since ratcheted down, so the standard mix that I'm meeting right now is the 97 PM2.5 in ozone. We're real close on the 2008. There's not been an adjustment since then. There'll probably be one. But our SIP, our state plan to meet it, is on a glide path of this time next summer, I will have Cleveland meeting the air quality standards. Now the unfortunate thing is between now and next summer, the federal government's going to lower ozone again. So right now it's at 75, it's going to probably drop to 70. I'm at 74. So I'm at 74, meeting 75, PM2.5, I'm real close. That's the glide path, that's the long pole in the tent, and we will meet that by next summer. But Cleveland will meet an established air quality standards, and nobody would ever thought we would be there. And we're there, which is exciting. That's exciting and that's really the take home. We have a long way to go. Look out at the lake. Absolutely no doubt. Lots of science needs to be done. But never lose sight of where we came. You know, there's a lot of waters out here now that you can wade in, where historically you waded in, you probably died. So there's most waters out here you can feel comfortable wading. Not all of them can you drink yet, but you can sure wade through them. So you can be fully submersed in them, you can swim in them, and everything other than drink, and there's only a handful that don't make the drinking standard. And those are traditionally some of my old abandoned mine sites, and the only issue there, the water's real clear. It just has a pH of 2. There's a little issue there. But you pH adjust it, and it's potable. So you just got to get past the pH dip. So a long way. So a lot of you, as you work through your decisions on going to school, continuing your education, a lot of us on this side of the room already did that. So we've been there, done that, as you heard me say earlier about a t-shirt. So we've made that choice. We made that choice because it wasn't that we didn't have other choices. And most of us probably had multiple other choices. I know I did. So as you work through your career, you get to the fork, don't be afraid, take the fork. There's not a right or a left, there's not a right or a wrong. You get to a point, guide on yourself. Guide with your church, guide with your family, guide with your parents, guide with your friends. Oftentimes you're going to go wrong. That's really a good message. So I do want to open it up for questions. Oftentimes I get some really odd ones. Jeff, this is my 3rd year doing this. So this is more of a fun presentation, so it's been a little more rattling than some of my more technical ones that I have to give. So thanks for having me here today.
Thanks very much.
Questions for Director Nally?
Anything on? I know it's getting late, we're all tired. Yes, in the back, please.
So I apologize, it's a little off topic, but I'm curious, what sport did you play in your state?
That's a good question. Actually, that probably typifies my personality. I wrestled at state. I was a wrestler, so that's pretty much my personality. And, Jeff's heard it in the media. I'm one of those type of people, if it's 80 on one and I'm the one, put your money on me. Again, the wrestling mentality. Off topic, good question, though. Please, Chris.
So with the rivers, I think it was like 89% meeting attainment. Has the attainment designation changed along with that, or has that been stable for the last, since the 80s, or have we changed the attainment?
What's changed is what is in the makeup of what's sampled. It used to be when you looked at attainment, you looked at like E. coli, you looked at basic nitrogen, and if you met that number, then you were good. Now we look at several more indicators, so it's not that they've been ratcheted down, although a couple of them have come down slightly, at least on the drinking water side, but it's that we've added additional parameters. So not only have we done better, we've gone from 20 to 89, but the universe of what we were sampling when we weren't meeting them before versus what we're meeting now is tremendously large. Just the whole pool of protocol samples where, and think of what you do out here on your camp plan, your discharge here. It used to be you probably sampled for 4 or 5 items, now you're probably sampling for 15. So it's not that those 4 or 5 that you were originally sampling for have gotten more strict, we've just added more to the pool of what you have to sample. So to be at 89 and with the added pool shows phenomenal, because you're moving the ball. Great question, please.
So the Lake Erie was, I think, probably one of the greatest, environmental success stories in terms of how we cleaned it up. And I don't think the country has noticed yet, but we've obviously gone in the other direction. And I think from what we heard today, that there's a good chance we're going to get back to where, the good place begins. But how do we stay? What do we need to do as a state to stay there and not kind of have this reversal?
Well, here, and I've heard Hector, actually, Jeff make the comment. When you look at, it goes back to what we're measuring, by the way, but when you look at the improvements that the farmers made the first time back in the 90s, they virtually cut their total phosphorus usage in half. And oh, by the way, we're less than half now. So here we had an issue in the lake. We asked the farmers to cut their phosphorus use. They cut that phosphorus use in half. And we are today lower than that half. So then the question is, what's changed? So to answer your question. So if we've already reduced overall phosphorus in the basin by more than half and maintained it, and now we're going after lower levels than that. We've asked Scott's to do 0 on lawn care. We went to 0 phosphorus on detergents, 0 phosphorus on laundry soap, which tends to get things light and clean. So we've had to wait for technology to catch us. So we've done all those, which is why we've been maintaining that half or less. So now you have to look from a science standpoint, what are we doing different? We got millions and millions and millions of linear foot of field tile that's gone in. So we asked farmers, because the whole rule of thumb, and for those of us in school that took soil science, if you were controlling erosion, controlling the soil, you were controlling phosphorus. That's what we were taught. So if you went out there and put drainage controls in and all that, and soils didn't move from your farm, we thought phosphorus didn't move from your farm. But we were measuring total, not dissolved. So what's happened is now is people have gone to no-till so that we can reduce erosion. So that's good. They've gone to tile so they can get on the fields more timely. So it goes back to when are they going to put the fertilizer on the field. Fields are drier quicker, they can get out there. So then what else has changed? Well, now they're broadcasting because doing no-till. So that's now making that available. So what we need to revisit is, where did we evolve? So we already cut it in half, right answer. We already went and controlled soils, right answer. Now we need to look at different technologies on application, or control drainage tile. So that's some things that we're wrestling with now, and that's some of the OSU work with Mike Libby and some of the folks. But then we need to be also much more creative out of the box. We need to be looking, to get to your point, and how do we stay there? We need to continually look at the 4 R's, right rate, right time, right amount. So we need to question from a soil chemistry standpoint, what's the right makeup of the fertilizers? But also remember, we dealt with, back 40 years ago, we were starting to deal with the sewage treatment plants. So we were building basic treatment plants that would handle things typically without a rain event. Most of these have not been dealing with the added volume of water from the rain event, because they're on 20 and 25-year glide paths. So they're in various phases of it. So even though we drive down the street and see a wastewater treatment plant, and we see it operating, remember, after a rain event like yesterday, shit through the screen door, no pun intended. So we need to keep our foot on the accelerator to deal with those issues from a brick and mortar infrastructure, so that we can then get to where we need to be, and that's not having Lake Erie be a blue-green algae system. So it's not that we haven't been doing the right things, we haven't completed anything. There are very few CSOs in the Western Basin that are done. Where you go to the coasts, where there's been more focus on it, again, traveling a lot, you look at Boston. Boston, 0 discharge in the Boston Harbor on a rain event. 0 discharge in the Boston Harbor on a rain event. And you talk about a community that's 200+ years old, where Ohio's about 138. So they figured it out. Charleston, South Carolina, San Diego, a lot of those, they're ahead of us. And by God, we need to catch up in Ohio. We just allowed ourselves to get behind.
Scott, is some of that, when you go in like East Ninth Street in Cleveland right now, you can see all kinds of utility work going on. It looks like they're working on sewer kinds of things. Is that some of what we're seeing?
Yeah, that's actually the big tunnel.
Okay.
So when you go up like Euclid Ave, that's that 4.5-mile tunnel that's about the diameter of this room. I've been down through it. So what happens is you've got all the streets, when the rain hits the street and it hits that culvert, all that drops into the tunnel instead of going right out into the lake. And then that tunnel fills up during the rain event. And then after the rain event, they pump back out to the headworks of the treatment plant and meter it back through the wastewater treatment plant to be treated. So you don't end up with that first flush and then the bypass, because of that first flush hitting the city's sanitary line. It can't handle the combination of the sanitary and the storm event, so it bypasses the whole thing. If not, well, you can sit there and think, well, Jesus, don't make it happen. Director, tell them to stop it. Well, when I tell them to stop it, then it backs up in your basement. So then you got raw sewage from your neighbor, because I've not allowed it to be bypassed, pull the relief cork at the other end, so it backs up in the system. And when it backs up in the system, where does it back up? Basements. So then you got raw sewage in your basement. So do you want raw sewage in your basement, or do you want raw sewage in your river or lake? Ideally, we don't want neither. But we chose the lesser of the evils, and it ends up in the lake and river, which is what we need to address. So really, in a nutshell, we need to do brick and mortar. We've already cut phosphorus a lot on the farming side. Now it's fine-tuning practice. Measuring, modeling, monitoring, looking at makeup of fertilize, looking at that stuff, and we have the ability to do that. And we've got a great team at OSU and Heidelberg, got some of the best agricultural facilities in the country, actually in the world. We'll figure that piece out. It's the timing issue on dealing with the CSOs. Because when you look at glide paths, these are 20+ years.
Last question. Scott, sort of talk about the size, employees, Ohio EPA.
Great.
You've lost the number to retirement that just went on. So size, employees, opportunities for students because of retirements, and opportunities for students while they're students, maybe because of that, like internships.
Yep, no, perfect, and that's just celebrated our 40th birthday in October as a state organization. A lot of my employees have been here the whole 40 years. So once the PERS system made changes, the public retirement system, in the last budget bill, it went into effect in January, I lost 100 plus people in a matter of a couple of days, a lot of institutional knowledge. When I teed up our balanced budget, I only took 56 off the limit rolls from attrition. So there are openings, as you hear in maybe other states. Ohio, we actually are hiring. So I'm hiring people all the time, and I've got, as I showed on my map, district offices around the state with a central office in Columbus. So we're hiring. We hire in a basic entry science position, usually an ES2 or an ES3. So you're talking at science in your title, or a geologist or a chemist, but most of us in this room are scientists. But all these stuff that we have people doing now on the internship side, which is really what you want me to get to. I have 80 plus interns that are running around the state right now, wading through streams and rivers, and they're going to pick me up tomorrow night when I do some electrofishing or electroshocking at 10 o'clock. So I mean, and that's what my interns are doing now. There's 80 plus of those that are on the surface water side of the house. I probably have a total of 130 interns as a total. I have people that are interning in IT, the large IT program. I have people that are interning in law school, so they're with my 38 plus attorneys in the agency. I've got people interning in my accounting area. We can always use assistants in accounting, and people are trying to decide career paths in schools. And then, of course, on the science field, I've got folks that are on surface water. Like I said, that's the biggest program we have. But I've got drinking water interns running around the state, working with some of these churches, working with some of the school systems, so that we can improve their ability to meet water quality standards. Now's a good time this summer to do that. So we oftentimes tee up 4 or 5 interns with one of our, we call it a mentor program, with one of our mentors, and we work around the state doing a lot of sampling, which is a lot of what gets run through the lab.
Are those primarily summer programs?
Those are summer programs.
And those would typically be full-time college students in those while they're in college?
Correct.
And they're, in addition to the education opportunity, are they paid for that?
They're paid for that. And we've expanded it because of the changes of quarters to semesters at OSU. We've made sure that, one, we've extended that summer program to reflect that, and then we've also made it available to after they graduate. So oftentimes our students, several of you in this room, for example, are wrestling with graduation, and you're trying to make a decision whether what you're at that fork. Do I go in for graduate school? Do I go into private sector? And we allow a bridge of that where you can get some exposure, work on a research project, work on one of my TMDL sites, and work on that for a summer. You've already graduated, and you're trying to make a decision in that fork. Do I go to graduate school, or do I go into the business sector? So we've been trying to provide that gap. I have a chunk of the 80 on the surface water side that are in that gap. It just provides maybe a little more direction. So if they do decide to go to graduate school in August, one, they've probably already got a project in mind. They've probably already teed up with one of the professors, maybe even a year with a project in mind. Sure makes it more palatable to a school then, because they have a highly
More informed decision.
more informed, much more mature when they're having the dialogue. Think of how you would want somebody coming in your door, for example, Tom, want somebody that's mature, already got a good head on their shoulders, been there, done that, and have made the conscious decision other than just kind of meandering in there going, I think this is what I want to do. And it's hard for you to get geared up behind it. We get that. So we're trying to bridge that, and that's a great closing for that. So.
Thanks very much.
Thanks.
Rick. 4th of July on Thursday, so we're not back in here till the 11th. And on the 11th, our research lecture is going to be from Dr. Joe Conroy. Joe was a student, coming back. Joe was a student up here and a professor. You'll see him someplace over there. Got his PhD actually working on nutrient management and plankton management within fish hatcheries. Within a lot of work also right in Sandusky Bay. Right now he's working in the fish hatchery business. So the students who are on REUs working out in Sandusky Bay, Joe Conroy did a whole lot of that. He'll be here next week. research at Ohio State, and she'll talk about research opportunities and recognize that it's one of the top research institutions, top 20 research institutions in the country, and I'm pretty much amazed at the things that are going on in some of the laboratories. It's really impressive, and she's a lot of fun. She'll be similar to Director Nally. So thanks very much for coming. See you on the 11th.